risk side. The U.S. has focused more on biosecurity, on both biowarfare and bioterrorism, potential applications in synthetic biology. The conversation in Europe has been more about biosecurity, which some have dubbed bioerrorism, in contrast to bioterrorism. And here you're looking at everything from lab accidents to inadvertent environmental releases, to horizontal gene transfer. You know, microbes can actually exchange genes. If a microbe, either in the lab, I suppose, or in the wild, if it got loose, it could evolve and adapt, potentially. And then you have the problem of cultural and economic dislocation. This is the wormwood, or the Gleason example. The concern that groups like EPC have about this is that basically there are people in the developing world who are making money by producing artemisinin from wormwood. You're now dislocated. But of course, the benefits of the antimalarial drugs will also flow largely to the developing world. Maybe not to the same persons, but to also people who are at lower end of the economic hierarchy. A bit more about the biosecurity risks. It is important to distinguish between biowarfare and bioterrorism. Essentially, biowarfare is the construction of toxic organisms and delivery systems by state actors. Bioterrorism would be non-state actors attempting the same thing. Now, there are a variety of political defenses, political, diplomatic defenses that can be erected against both more effectively biowarfare because states tend to be more responsive to, particularly to diplomatic initiatives, but to some extent bioterrorism. There are quite significant scientific and technological hurdles posed by anyone who would attempt to do either biowarfare or bioterrorism with synthetic biology. For non-state actors, the consensus seems to be that the problem won't be so much, well, there will be a challenge even creating it, the organism they seek, but they'll have a hell of a problem trying to weaponize it. Weaponization turns out to be a very complex task. So maybe you can create a really nasty bug. You're going to have a hard time delivering it the way it's going to have the scale of impact that you wish to have. Sad to say, the technology that turns out to have the highest, two technologies that have had the greatest impact on terrorism were jet airliners and box cutters. There is in the U.S. something called the National Science Advisory Board for Biosecurity. This is from a recent report, this text. This is actually useful, I think, to read in its entirety. Current scientific understanding reveals that it is often the combination or interaction of genetic elements that underlie these properties. The properties here are essentially toxicity, infectivity, transmissibility, rather than one specific gene sequence. Furthermore, the harmful consequences of biological agents are dependent upon the coordination of multiple factors, including host susceptibility, the agent's infectivity, transmissibility and virulence, and the availability of prophylactic or therapeutic interventions. Now, I want to make a couple observations about this. This being largely bench scientists, they didn't mention public health strategies, which I think actually happen to be quite valuable. We know furthermore that nature is fully capable of devising lethal organisms without synthetic biology or genetic engineering. The current outbreaks of swine, so-called swine flu, are ample evidence of that. There are suggestions, and I don't know where today's news is on this, or what we will know 6 months or a year from now. There are suggestions that human practices may in fact have encouraged the development, or at least the spread of this. There's been the accusation that maybe some practices of large-scale mega pig farms might have had something to do with either the evolution of this particular strain or with the spread of the bacteria, the virus. So my conclusion here is that complexity and emergent properties may be our friends in this case. It's just very hard to make an organism that will be lethal. quite frankly, the only way you're going to know that it's harmful and transmissible in humans is to make it and then try it out on humans. So I'm not sure who's going to be on the research ethics committees that look over those trials. So there are a number of things we can do in terms of public policy regulation. The primary ones are self-governance, government regulation, and international agreements. There was an interesting little case study of using self-governance among scientists. Now, you'll be shocked to know that generally scientists prefer self-governance. I'm sure that comes as a great surprise. The American Society for Microbiology did a little test in the year 2003 where they decided they would review all the manuscripts submitted for publication in their journal. And they would see if they could identify those manuscripts that contained information that was potentially, had great potential for misuse. So they screened more than 16,000 manuscripts. Only 3 were identified as needing any kind of special attention, and only 1 had any modification. And in the end, all three were published. So, what's the lesson from this? I mean, you could say that, well, there's really not much there to be worried about, or you could say that self-governance may not be as powerful as we'd like it to be. And I leave it to you to decide what the lesson is. I'm not sure. But it resulted in a lot of effort, very, very little impact and change. Now, Don Kennedy is, I've had the good fortune to know Don quite well. He's the former president of Stanford, the former editor of Science. And he said this some years ago. When did biology become big science? He said, when the capital cost of the equipment and special facilities needed to support a scientist becomes larger than the capital value of the endowment necessary to yield a salary. When did this happen? He dates that particular Rubicon to the 1970s, well before the even genome project. The irony here is that it may be that synthetic biology, at least strains of biology, will once again become small science. In fact, it may deal with the phenomenon of what's been called garage biology, on the analogy with the garage computer movement